The urinary system determines what is filtered and retained in the blood. These kidneys are showing how the red particles are too large to be filtered out, therefore they are being retained in the body. During filtration, some things are filtered and some things are retained. Things that are filtered include glucose, ions such as salts, water, amino acids, hormones, vitamins, and plasma without proteins. Things that are retained include albumin, red blood cells, white blood cells, and platelets. Filtration starts in the renal artery. It travels up the interlobular artery, goes around the arcuate artery, through the nephron, down the renal medulla into the renal papilla, into the minor calyx, in the major calyx, the renal pelvis, and out the ureter. Retention begins in the renal artery. It travels up the interlobular artery and around the arcuate artery, down the nephron, which is reabsorbed, and travels through the arcuate vein, down the interlobular vein, and out the renal vein. This is the nephron, which is the functional unit of the kidney. It filters blood, removes toxins, and functions in osmoregulation. The afferent arteriole receives blood from the kidney to begin glomerular filtration. It is wider than the efferent arteriole in order to maximize the filtration rate. The glomerulus is where filtration actually occurs. It decides whether things are filtered out of the kidney or retained in the blood. Bowman's capsule surrounds the glomerulus and contains filtrate fluid. Together, the glomerulus and Bowman's capsule make up the renal capsule. The efferent arteriole takes substances to be retained within the body to the blood vessels. Following the glomerulus is the proximal convoluted tubule, the loop of Henle, and the distal convoluted tubule. Together, these make up the renal tubule, which functions in reabsorption. The peritubular capillaries surrounding the renal tubule are the receptors for substances being reabsorbed. The renal tubule leads into the collecting duct, which carries filtrate to the renal papilla. The arcuate artery makes the border between the cortex and the medulla.
No, not that kind of stoned. As you can see, this hippie is drinking soda and eating chips. The high salt and dehydration occurring in her body increases the chances of kidney stone formation. The first step in kidney stone formation is called nucleation. The first kind of nucleation is homogeneous nucleation, which is when a crystal nidus forms with two of the same ions. The second kind is heterogeneous nucleation, which is when a crystal nidus has a composition consisting of different ion types. During the second step, growth, the crystals grow in size. This happens at the renal papilla. The third step is aggregation. Here the crystals spontaneously stick together. Retention is when the stones remain on the medullary wall and can grow for a length of time. It is still unknown as to why this occurs.
There are 5 different kinds of kidney stones: uric acid, cytosine, calcium phosphate, struvite, and calcium oxalate, which is the most common.
Kidney stones spontaneously detach and move through the minor calyx, major calyx, and out at the renal pelvis to be removed via the ureters.